Hey everybody, Mr. J here, and today we are going to be talking about multiple alleles, and I'm specifically going to use the example of blood type, which is really helpful primarily for your classes that you're taking right now. So, what are multiple alleles? Well, remember alleles is a variation of a gene. So a gene is something that codes for a trait, right? So DNA that codes for a trait, but then those traits can be variant, right? We can have multiple. So for example, say there's a gene for eye color. Well, color, you can have all sorts of variations of color. The different variations would be the alleles. So the definition of multiple alleles is when one gene has more than two variations, more than two alleles associated with that gene. So I'm going to use the example of blood type. So if you were to look at every person in the world, okay? If you look at their chromosome number 9, there are 3 possibilities of blood types on those chromosomes. So let me explain further. If you look at each chromosome 9, I have a different little color on each one. And each different color is basically designating which allele is being coded for within that gene. So this region on all those chromosome 9s contains that blood type gene. But there are 3 variations in the human population. So what do they code for? Well, if it's black, I'm just using this as an example, say if you have a chromosome 9 that looks like this. Well, this is going to code for the let me come down here. The antigen, which is just a protein, A. Okay? So antigen A is basically a protein that sits on the outside of your blood cells. And it's basically just saying, hey, I have this flag called antigen A. I belong to A. I love A. So it's a protein antigen A on the outside of your red blood cell. So if you have this specific allele, this specific variation that codes for an antigen A, you will have A antigens, okay? What if it's blue? Well, if you have a blue chromosome, a blue allele on chromosome 9, we're going to say this is antigen B. So in this example, you are going to have a blood cell, and you're going to have antigen B. So little flags saying, hey, I am B. If you receive the last one, which we're going to designate as red, this designates no antigen. Okay? So if you have no antigen, so you have the red, you are going to have a red blood cell with no flags. So there we have it. We have an allele coding for A protein. We have allele coding for B protein, and we have an allele that doesn't code for a protein, so the lack thereof. So, remember, in your cells, you have 2 of each chromosome, right? You have 2 chromosome 1s, 2 chromosome 2s, and so forth. So therefore, you have 2 chromosome 9s. So you take your pick. You get 2 of these, all right? You can have 2 of these, 2 of these, 2 of these, or you can have a mix match of them, okay? So maybe you have 1 red, 1 blue. Maybe you have 1 red, 1 black. Maybe you have 1 black, 1 blue. It's just a mix match, and this is where we determine our blood types, okay? So let me explain really quickly. When we have these antigens, we are going to code for them using alleles, and the way we write them is usually with a big I with a big A for antigen A. So that will be the allele for antigen A. For antigen B, we use big I, big B. Okay? Why am I writing them as capital letters, do you think? Because they're both dominant, right? So they're going to show codominance here in a second. Whereas if we have no antigen, we're actually going to write this as little I. It is recessive. Interesting. Okay, so let's talk about possibilities. Well, let's say we have a genotype where you have big I, big A, big I, big A. What do you think that's going to code for? So basically, you just received 2 of these guys, right? Well, you're going to have type A blood. But you could also have big I, big A, and a recessive. And what would you still have? Well, you would still have type A blood. So you would still have antigen A because it's dominant. You have a recessive with no antigen, therefore you are type A, okay? What if, for example, you were big I, big B, big I, big B? Obviously type B, you had, let me actually draw that in blue so I can keep it steady. Let's say we have two of those, big I, big B. That's obviously going to be type B blood. And in the same vein, say you're big I, big B, but then you had a little I. Well, regardless, what type are you still going to be? You're still going to be type B. Okay, because even if you have a big I, big B, and a little I, you're still going to show that antigen B. So you have type B blood. What if you had a big I, big A, but then you received big I, big B? This is a tricky one. Well, you received a big I, big A. That means you're going to show this A. You received a B. That means you're going to show the B. So, for this example, you're going to be type A, B. Type AB blood. Okay, last one. Say you have two little I's. This is the only example where you will have type O blood. So this is type O. So if you receive two of these reds, they're both recessive, it's going to show type O. So in other words, you have no antigen. Well, the only one I didn't show you on here is, let's say you have type AB. Well, in this example, you would have to receive IA, IB, and this would show up like this. You'd have a blood cell, and you would have both A, and you would also have the B antigen as well. So that would be type AB. But remember, you inherit two of these, so you kind of can mix and match. So these are all the possible genotypes coding for the phenotypes, the actual physical trait of that red blood cell. Awesome. So let's get into some examples. Let's say you have a parent. I'm going to erase this a little bit. Let's say you have a parent that is type A blood, and they have big I, big A, big I, big A. Let's say you cross it with a parent who has O blood. So this is parent 2, both little I, type O. Parent number 1, type A with both dominant A's. Well, in this case, if you cross all of them over, each and every offspring will have the A antigen, yes? And also, you're going to have a recessive one, right? But for this example, all of them are big I, big A, little I. Therefore, all of them are going to show type A blood. So you could say 100% of the phenotype would be type A. Awesome. Now let's do a cool example. All right, let's say you have a parent that is big I, big A, and little I. Then you have another parent that is little I and big I, big B. Well, now let's do this really cool cross. So I'm basically saying your mom, for example, is still type A, right? Because she's got big I, big A, and a little I, showing that A antigen. Dad is type B blood because he's got that B antigen, but then a recessive not showing up. But offspring, let's check it out. The first offspring, excuse me, would be this. They would be big I, big A, big I, big B. That would make them type AB. As we continue on, what gets even cooler is we're going to show all of those genotypes. So check this out. We've got big I, big B, as well as an I. We've got a big I, wait a second, big I, big A, as well as a little I. And then we've got two little I's. So if we look at this cross, we show up every single possible phenotype actually, but we all have different genotypes. So this first one would be AB blood, second one, B blood, third one, A blood, and fourth one, O blood. So the way we could write this is that 25% all around could possibly be type A, type B, type AB, or type O blood. So there'd be a 25% chance for that offspring to basically be any blood type. Awesome. So this was multiple alleles. I kind of brought in some codominance here too, because we see that the two dominant alleles both fully show up. And we also had multiple alleles because we have 3 possible alleles we are dealing with today. So hopefully this is helpful. This was ABO blood typing. If you have any questions, please drop them in the comments below. Be sure to like and subscribe if this is helpful. And thank you so much for watching.